Hello, and welcome to our Sample Tech Talk. My name is Harris Chowdhury. I'm the Product Marketing Manager here at Sampled, and today I'm joined by Dr. Michael Sheldon, who is the Senior Director of Scientific Affairs here at Sampled. Hi, Mike. Why don't you get us started by telling us a little bit about yourself and your role here?
Hi, Harris, and hi, everyone watching. My name is Mike Sheldon. I joined Sampled after more than 20 years of experience as a bench scientist in genetics and neurodevelopment. And I've now been with the company for 16 years, and during that time have overseen the operations of our sample processing laboratories. The breadth of my experience, both prior to as well as after joining Sampled, led to my being tasked with founding a scientific affairs department. Scientific Affairs has as its central mission at Sampled to provide technical resources for our clients and to act as a liaison between them, our laboratories, and our commercial team to ensure that our clients get customized solutions to enable success in their research.
And we're here to talk about single cell RNA sequencing. Could you tell us the difference between single cell RNA sequencing and bulk RNA sequencing? And then tell us a little bit more about how single cell RNA sequencing works.
Sure. So let's start with RNA and the transcriptome. The development of a comprehensive catalog of the expression of our genes encoded as chromosomal DNA to give rise to RNA, central to so many biological processes, is of fundamental importance to the study of human disease and development. Now, the collection of RNA molecules expressed at a given time and in a given cellular environment is called the transcriptome. The first methods that were developed to study the transcriptome involved the use of microarrays, substrates onto which homologous probes have been affixed or arrayed. Extracted RNA from cells or tissues is then hybridized to the substrate, allowed to bind, and then those hybridization events give rise to a quantitative signal that correlates with the expression of that gene in that extract. Now, microarray technology is very powerful and has yielded many important results, but is largely now being supplanted by new next generation sequencing based techniques to interrogate the transcriptome. The first of these, RNA sequencing or RNA-seq, takes that same extracted RNA and reverse transcribes it to generate cDNA libraries, which are then sequenced using any number of technologies, such as those of Illumina or PacBio, to name two that are in use in our laboratory. RNA sequencing offers significant advantages over microarrays, including the ability to transcend defined probes on the arrays and identify both known and novel features in a single assay, giving insights into transcript isoforms, gene fusions, and single nucleotide variants, to name a few. Now, as powerful as they are, both microarrays and RNA sequencing are so-called bulk approaches, limited in their ability to resolve expression differences between single cells or between adjacent regions of a tissue, such as a tumor. These differences are fundamental to our understanding of biological processes as diverse as the development of the brain or complex interactions between tumor cells and their microenvironment. The advent of single-cell RNA sequencing has armed researchers with a precise tool for analyzing the transcriptome at the level of a single cell, even among tens of thousands of cells in a given population. Think of it as the difference between wanting to study how a car works by looking at the average of all the cars on the LA freeway versus being able to compare a single Ford to a single Ferrari and look at their differences and similarities. Now, how does RNA sequencing at the level of single cell make this possible? Well, a number of breakthroughs and improvements in processing have led to this. Protocols were developed for collecting and processing tissue samples to capture single cells and minimize their aggregation. This allows for the creation of reverse transcribed libraries with barcoded molecules, so with small molecular DNA tags affixed, that can then be subjected to sequencing and bioinformatic analysis. Now, the barcodes are the key to the ability to resolve at the level of single cells. The large number of diverse sequences in the barcoded library provides unique tags for individual cells. These come through in the sequencing data and can be resolved using bioinformatics approaches to give a unique transcriptomic profile for each individual cell.
Great. Can you tell us a little bit about the applications of single cell RNA sequencing
Mhm.
too?
Oh, there are a great many. They span virtually all fields of biomedical and basic research. I touched on tumor biology before. The ability to generate gene expression profiles for individual cells gives the pathologist an unprecedented ability to classify tumors at the molecular level and allows us to look at things like gene expression changes during tumor progression or response to therapy, or how the tumor microenvironment influences tumor growth and regression. Developmental biologists can identify whole new cellular subtypes and examine their roles in normal and disease states, for example.
Great. And can you tell us some of the advantages of
hmm
outsourcing single cell RNA sequencing to a company like Sampled?
hmm Oh, there are a great many advantages to using a company with a global integrated analytical biorepository such as that at Sampled. Sampled has decades of experience with hundreds of thousands of samples in all aspects of biosample collection and processing with the ultimate goal of genomics applications. And this is in our College of American Pathologists and CLIA certified laboratories. These services include the extraction and quality control of nucleic acid to ensure the highest yield and quality of samples for genomic analysis, such as single cell RNA-seq. Further, as a certified provider of 10x genomics services, including Chromium single cell RNA-seq, our clients can be confident that their samples will yield reliable and reproducible results in support of their goals for clinical trials or basic research projects. We are working towards adding spatial transcriptomics services at both the cellular and subcellular level later this year. These incredibly powerful technologies will enable researchers to pinpoint transcriptome-wide gene expression to specific cells and tissues with insights into their spatial relationship to one another within the context of the tissue, and is emblematic of Sampled's continual effort to provide new technologies for our clients' benefit and stay ahead of the curve.
Great. Thank you, Mike. And thank you for joining us for this Sampled Tech Talk. We hope to see you at the next one. Thank you and goodbye.
Thank you.